students, welcome to Sunil's tutorial. I am Sunil Melwani and today we will be doing this chapter called as Genetic Basis of Inheritance. Let's see the reasons for Mendel's success. Reasons for Mendel's success. Reasons for Mendel's success. First, now, the pea plant that Mendel chose for conducting his experiments was most ideal for controlled breeding since it could be easily subjected to cross-pollination. Right? In order to know the results, you need to do a cross-pollination, and this plant could easily be subjected to cross-pollination. The pea plant which Mendel chose for conducting experiments is most ideal for controlled breeding is most ideal for controlled breeding since it can easily be subjected to cross-pollination. Since it can easily be subjected to cross-pollination. Right, that was the first reason for the success of his experiment. Next, he clearly identified the contrasting characters. He was very clear about the contrasting characters of the pea plant. He identified very clear contrasting characters of the pea plant. Right? Then he made sure that he selected pure breeding plants for his experiments. He is said to have spent 2 years to ascertain his characteristic feature. He selected pure breeding plant. For his experiment. Okay. Then he concentrated at a time only on one particular trait, which the two contrasting he concentrated on only one particular character at a time while performing the experiment. See, he would take a trait and he would find out about its contrasting conditions. Instead of trying to check for all the 7 different characters, he would concentrate on one character at a given period of time. Right? He concentrated at a time only on the inheritance of one particular trait. Instead of attempting the inheritance of the entire set of characters. Instead of attempting the inheritance of entire set of characters. So at a time he would select one character and he would just concentrate on that. He maintained very accurate records of all the observations that he had made. He maintained accurate He maintained accurate records of all the observations. He maintained accurate records of all the observations he made on the breeding experiment. Observations he made on the breeding experiments. He maintained accurate records of all the observations he made on the breeding experiment that he had designed. Then he pooled the data obtained from similar experiments into different characteristics and analyzed the result. Whatever similar answers he would get, he would pool that data together. How he has got those similar answers? So he pooled the data obtained from similar experiments. He pooled the data obtained from similar experiments. He pooled the data obtained from similar experiments for different characteristics. Different characteristics and analyze the results. And analyzed the results by using statistical method and applying the law of probability. By using statistical method, and applying laws of probability. Applying law of probability. Right? Then he was able to effectively check the flower under investigation from contamination by unwanted pollen grain. Remember, I just showed you his experiment. He would emasculate the flower and then he would cover that flower with a plastic bag so that no foreign pollen grain would fall on it. Right? So that was one of the reasons for the success of his experiment. He was able to effectively check. He was able to effectively check the flowers under investigation. The flowers under investigation. He was able to effectively check the flowers under investigation from contamination by unwanted pollen grains. Fine. And he was fortunate enough for choosing the 7 pairs of contrasting characters in the pea plant. It was later discovered that the gene responsible for these characters are located in were located in separate chromosomes. So it was very easy for him to get those results. He was fortunate enough in choosing the 7 pairs. In choosing the 7 pairs of contrasting characters. He was fortunate enough in choosing the 7 pairs of contrasting characters in pea plant. It was later discovered that the gene responsible for these characters It was later discovered that the gene responsible for these characters are located in separate chromosomes. Are located in separate chromosomes. Right? And finally, he was fortunate in the sense that the characters that he had chosen in the pea plant did not show any interaction or linkage. He was also fortunate the two characters didn't mix with each other. Right? He was also fortunate. He was also fortunate that the characters he had chosen in the pea plant, that the characters he had chosen in pea plant, the characters that he had chosen in pea plant did not show any interaction, did not show any interaction or linkage. So those were the reasons for Mendel's success. Now let's start doing his experiments. We've seen the procedure of his experiments. First, let's do his experiment of monohybrid cross. Now we said that he would do one character at a time, right? So let's see how he did monohybrid cross experiment. Monohybrid cross experiment. Cross experiment. Now we already know the procedure of his experiment. What he would do is first get the pure parent, then he would do a cross between a pair of contrasting characters of pure parents, and then he would do selfing. Right? Now first of all, what do I mean by monohybrid cross? Monohybrid cross is a cross between two pure parents in which inheritance pattern of only one pair of contrasting characters is studied. Out of the 7 characters, I am going to be studying only one pair of contrasting characters. All the other 6 pairs are going to be kept same. It's only one pair that would be studied. Right? So what is monohybrid cross? Monohybrid cross, a cross between two pure parents, a cross between two pure parents, a cross between two pure parents in which inheritance pattern, of only one pair of contrasting characters, is studied, is called monohybrid. It's called monohybrid cross. Fine, we get this in here. So in monohybrid cross, it is a cross between two pure parents differing in only one single pair of contrasting characters. The remaining other 6 characters I will keep same, only one character would be different between the two parents, right? So I could say that it is a cross between two pure parents differing in a single pair of contrasting characters. In a single pair of contrasting characters. Right? That is what is monohybrid cross. Now, how did he do the monohybrid cross experiment? That means out of the 7 characters that he was studying, he would keep 6 characters same, and only the 7th character would be a contrasting character. Right? Step 1. The normal steps that he had followed in doing the experiments. Right? First, Mendel first selected a pure tall pea plant as a female plant and a pure dwarf pea plant as a male plant. Mendel first selected a pure tall pea plant as a female parent and a pure dwarf pea plant as a male parent. Right? So he selected this. He confirmed that they were true breeding by selfing them for 3 generations. He confirmed that they were true breeding. By selfing them for 3 generations. By selfing them for 3 generations. Right? So that was the first thing that he did when he wanted to do a monohybrid cross experiment. Second step. So now by doing this, he's got the pure parent. Once he has the pure parent, step 2. Step 2. Now what did he do in step 2? He performed an artificial cross and collected the seeds and sowed the seeds. Same the normal procedure that he is following. He performed artificial cross, collected seeds, and sowed the seeds. Right? Now, he found that all the plants that he got were tall. He found that all the plants of F1 generation were tall. Right? Now, then step 3. He allowed natural selfing of all the hybrids and collected the seeds again and sowed them. Right? He allowed natural selfing of all the hybrids. Collected the seeds and sowed the seeds. Right? He found that dwarfness reappeared in some plants. Right? Now, in all he collected 1064 seeds of the F2 generation. In all he collected 1064 seeds of F2 generation, right? In which he found that 787 were tall and 277 were dwarf. Of these, 787 grew tall and 277 grew dwarf. Now, in order to make sure that this was not by chance, what he did was he performed a reciprocal cross. What do you mean by reciprocal cross? Like when he was doing the experiment first time, he took a tall female and a dwarf male. Now he did the same experiment again, taking a tall male and a dwarf female, right? Then he then performed reciprocal cross. He then performed reciprocal cross, and that is, he started with a tall plant as male parent and a dwarf as a female parent. He started with a tall plant as male parent and dwarf plant as a female parent, right? He observed that he got similar results. He got similar results. He got similar results. That is all tall in F1. All tall in F1 and tall to dwarf in approximate 3:1 ratio in F2. Right? So this was his monohybrid cross experiment. Right? In this manner, he did this monohybrid cross and reciprocal cross experiment with all 7 pairs of contrasting characters separately and obtained similar results. In this manner, he did monohybrid cross. He did monohybrid cross and reciprocal cross and reciprocal cross. He did monohybrid cross and reciprocal cross with all 7 pairs of contrasting characters. With all 7 pairs of contrasting characters and characters separately and obtained similar results. That is similar results, that means he got the ratio as 3:1. So your monohybrid cross ratio was 3:1. So do we understand the monohybrid cross experiment? Now that we know the monohybrid cross experiment, let's have an explanation of the monohybrid cross and the ratio with the help of a checkerboard. Monohybrid cross and ratio with the help of checkerboard. Monohybrid cross and ratio with the help of checkerboard. Right? Let's see this. Now, he took, let's see this, parent phenotype. parent phenotype, he took a pure tall and pure dwarf. Pure tall and he crossed it with pure dwarf. Right? So pure tall, what will be the genotype for that? Pure tall will be TT. Pure dwarf will be nothing but TT. Right? Now this is going to produce gametes. So the gamete that I will get, obviously I will get identical gamete, that is T. From here I will get a gamete of T, and from here I will get a gamete of small t. Right? Now, from where he got the F1 generation? The F1 generation would have one T gamete and one small t gamete. So this was my F1 generation, which was hybrid tall. Hybrid tall as in physically it was tall, but it did not have homozygous chromosome, it had heterozygous chromosome. Right? Now this hybrid tall now would produce two different types of gametes. Since this is hybrid tall, this would produce this here and here. This was the cross that was done that gave me hybrid tall. Now this would either produce a gamete of capital T or produce a gamete of small t. Right? Now you had selfing now. This was parent generation, F1 generation. Then F1 generation was allowed to self. So here you will have selfing. Right? That means what is selfing? A hybrid tall, that is TT, was allowed to cross with another hybrid tall, that is TT. Right? So let's make the checkerboard pattern. You would have male, female, male, female. Right? Now, if I take male on the x-axis, so that will give me T and T chromosome. Female on the y-axis, T and T chromosome. That's my checkerboard pattern. Right? So if I have a cross between them, now let's see what are the genotypes I would get. If I have a cross between this male and this female, this will give me TT. This male and this female, sorry, this male and this female will give me TT. Male, female will give me TT, and male, female will give me small TT. So that means this is going to give me a tall plant, pure tall plant. This is going to give me a hybrid tall plant. This will also give me a hybrid tall plant, and this will give me a dwarf plant. Right? That's my F2 generation. So in F2 generation I would get 3 tall plants and 1 dwarf plant. This is my checkerboard. Fine, do we get this thing here? So, in which case I can now say that my phenotype ratio is 3 tall and 1 dwarf. That is 75% is to 25%. So 75% I would get a tall plant, 25% I would get a dwarf plant. Thus, the F2 generation consists of the plants. F2 generation consists of the plants. 1 tall tall homozygous pure. That was my TT. So there are 25% of the chances that I'll get one tall homozygous pure. Then I get two tall but they are heterozygous. That is PT, right? So 50% chances that I'll get a heterozygous, and I get the third plant that I'll get is a dwarf, which will be homozygous, right? Again, this is homozygous pure TT, 25% chances. So that's what I'm going to get. So my genotype ratio is going to be pure tall, hybrid tall, and pure dwarf. This is 1 TT, 2 TT, and 1 TT. That's going to be my monohybrid cross and ratio with the help of checkerboard pattern, right? Now, let's try to explain why we have something like this. In monohybrid cross of true breeding homozygous tall plant with true breeding homozygous dwarf plant, let's see what happens. In Mendel's monohybrid cross, a true breeding homozygous tall plant, a true breeding homozygous tall plant TT is crossed with a true breeding homozygous short dwarf plant TT. That's his experiment. Now when these plants are crossed by hybridization, the plants belonging to F1 generation are developed. When these plants are crossed by hybridization, that is your artificial pollination, hybridization. When these plants are crossed by hybridization, the plants belonging to F1 generation, are developed or were developed. Now in F1 generation, what was observed were all were tall. Right? Now, this was my F1 generation. I've already shown you this with the help of the diagram. So you had pure parents, gametes, from where you had pure parents which gave you gametes that gave you the F1 generation. In F1 generation you got hybrid tall, that is all the plants that were obtained here were tall. Okay, done. Now let's move on to the F2 generation. How did I get the F2 generation? You had selfing between the F1 generation. So you had these F1 plants were inbred again. These F1 plants were inbred again, and the next generation F2 was raised, and the next generation F2 was raised. So I got F2 generation. Now in F2 generation what happened? In F2 generation two types of plants were obtained, tall and dwarf. In F2 generation two types of plants were obtained, tall and dwarf, in the ratio of 3:1. Right? Now, when selfing takes place, if I do this selfing using checkerboard, each type of male gamete had equal chances to fuse with either T, capital T, or small t of the female gamete. So a possible combination of three genotypes was obtained. TT, that's capital TT, capital T, small t, and small tt. Right? When selfing takes place, if you had selfing takes place, each type of male gamete had equal chance to fuse. Each type of male gamete had equal chance to fuse with. Each type of male gamete had equal chance to fuse with either T or small t of the female gamete. Right? So a possible combination of three genotypes was obtained. A possible combination of three types of genotypes was obtained. There were three types of genotypes. One was TT, that is pure tall. Then you had 2TT, hybrid tall. And the third was 1TT, that is pure dwarf. Right? So thus you had the phenotype ratio of 3:1. Thus phenotype ratio of 3:1. But what will be the genotype ratio? Genotype ratio is 1:2:1. One pure tall, two hybrid tall, and one dwarf. Right? So, thus the results indicate that even though the regressive dwarf character is not seen in the F1 generation, but it reappears in the F2 generation. In F1 generation, you had all tall, but in F2 generation, the character reappears. The results indicate that even though the regressive dwarf character was not seen in F1 hybrid, but 25% of the F2 generation had regressive factor in pure form. 25% of F2 generation had recessive factor in pure form. Right? This indicates that the factors maintain their purity, and as the gametes always received only one factor of the character. So this is my entire experiment of monohybrid cross, how I would get different ratios using monohybrid cross. Right? We'll stop this here for the day. Thank you very much.